Hello students. Today I am going to discuss about the third part of molecular breeding for crop improvement. In the previous classes, I discussed DNA fingerprinting, the marker assisted selection and the backcross, and also the marker assisted pyramiding. In this class, I am going to discuss the QTL mapping, which is a little bit important portion, and it is also very tricky part of this chapter. And so I suggest you that listen carefully, because if you do not give the attention, then this part remains not understanding for you. So let's start. What is QTL? QTL stands for the quantitative trait locus, and this is from the name you can understand that it deals with some quantitative data. And what is a quantitative data? That means the data what is countable. In case of plants, the quantitative traits means those traits, those expression of genes we can measure by means of any digital form. It is the analysis that deals with some statistical methods which links between the phenotypic data and genotypic data. The phenotypic data, those are polygenetically controlled, are calculated by quantitative trait locus. And only the quantitative data can be analyzed. We cannot analyze any qualitative data. That means if the fruit will be sweet or not, and how much sweetness is there, we cannot calculate this. But we can calculate the coloration, degree of coloration, the height, the kernel shape. Those can be measured easily, and we can calculate these phenotypic data using quantitative trait locus mapping. It involves the DNA testing, DNA marker testing throughout the genome for the likelihood that they are associated with a QTL or not. The principles of QTL mapping genes and markers segregate via chromosome recombination during meiosis, thus allowing their analysis in the progeny. The linkage means the chance of segregation. Whether two genes are linked or not, that depends on the percentage or degree of their segregation. Let's take an example. Suppose the yield of rice and the anthocyanin pigmentation at the leaf of rice base are two linked markers. This anthocyanin pigmentation is the marker for the yield of rice. Now, these two genes can co-segregate during the meiosis. If the genetic distance, the chromosome, the distance between two genes are more, then the segregation percentage reduces. Suppose gene A and gene B are located at a distance of 1 centimorgan, the chance of their co-segregation is different with the chance that one gene is A gene is located at a distance of B gene, that is 5 centimorgan. The chance is much lower here. The distance is the more the distance, the less the chance of co-segregation. So it depends on the lot score, which I am going to discuss later. So suppose in case of quantitative trait loci, we we deal with various markers, and I previously discussed there are various markers. One is phenotypic markers, the molecular markers, the biochemical markers, etc. Now take an example. If there are 4 plants, 1, 2, 3 and 4, and there are several plants here, and the number of plants are different. These plants are plotted in the same plot, and these are the same species of plants, but their varieties are different. Now we are searching for the salt tolerance property of the plant, and we found that in a natural condition, plant 1 and plant 3 give the salt tolerance property, and they are relatively higher salt tolerant than other the plants, and this salt tolerance is acting as the phenotypic marker. Now if we extract DNA from these plants and we run the gel, then the DNA bending pattern can be achieved in the gel, agarose gel, and this bending pattern is known as the physical map. This is the physical map where we can found that one or two bending patterns are different compared to others. Here the plant 1 and plant 3, which I found that these are salt tolerant, relatively salt tolerant than the plant 2 and 4. They produce the two unique bands in their profile, in their bending profile, or in their physical map. So I can uh, tell that these are the molecular markers for salt tolerance, and these may not be, but I can tell that these are now a assuming part of the next portion, next experimentation process. So we can tell that these bands may have some roles in salt tolerance. Now if I extract the enzymes from all those plants, and we found that enzyme A, which is produced by both of the plants, and this enzyme A is previously reported that it has some role in salt tolerance, and now we are searching that this enzyme A is linked with those molecular markers that I have found in the previous gel section or not. Now for this I run a I design an experiment that the comparison between the chemical markers for enzyme A and the DNA markers for the molecular markers, these and these. So I plotted these markers in the x-axis for DNA markers, molecular markers, and in y-axis the biochemical markers are plotted. And for all the plants, the data is plotted in this graph, and this seems like this produced the map like these. The graph will represented like this, and if I plot by if I analyze these data with some statistical process known as the regression analysis, then I can found either these or can the or these or various types of regression data can be found, and from those regression data we can correlate the chemical markers and the molecular markers. So this is the step, a key step for a quantitative state locus mapping. And using these data, we can easily locate the molecular markers for the salt tolerant gene at the chromosome, on the chromosome, and this is the map on chromosome for the molecular markers for the salt tolerance gene. This map is known as QTL map. Okay, if I am considering here the one chromosome, and for rice plant, suppose there is 36 number of chromosome, we can generate a huge map which is related with the various type of quantitative traits. Here I took the example of salt tolerance. This can be samely applied for the other various type of stress, like drought tolerance, like insect resistance power, like the yield, like the rice grain length, etc. Various those can be mapped by this technique. This is the basic principle of QTL mapping. Now come to the point, that is Lord score. I previously told that lord score is the acronym of logarithm of odd. In genetics, the lord score is the statistical estimate of whether two genes are likely to be located near each other on a chromosome and therefore likely to be co-segregated. A lord score of 3 or higher is generally understood to mean that two genes are located close to each other on the chromosome. Suppose, take the example, these two genes are placed in this distance and they are called at the link genes because they can segregate. There is a chance of the segregation together of for these two genes. But if these two genes are located in different chromosomes or in the same chromosome on different arms, if the distance between these genes are much higher, then we cannot say that these genes are linked, but we can use the term for as a non-linked gene. We can term this as the non-linked gene, and this linkage is can be calculated by the lord score value. The more the lord score, the chance is much higher for the co-segregation, and this can be plotted in this map, in this graph. And if we calculate the peak, this is the peak, and I can say that these point can be co-segregated for these DNA markers and these biochemical markers, and this gene is highest value of lord score. So what are the total process? First, we from the genotype we can calculate the genotype probabilities, and after calculating this, we can calculate the kin matrix, which I already discussed. The kin matrix means the probability matrix of two genes present, and this is the possible map actually. And there are various phenotypes and physical maps are there, and we can link between the genotype and phenotype. This is the basic principle of QTL, and perform a genome scan and find the lord peaks, and after that the permutation test was performed, and after that the QTL effects are estimated, and the short nucleotide polymorphism or the genetic molecular marker are identified in a quantitative trait loci map. So what are the significances? The QTL allows researchers in field as a diverse in agriculture and evolution and medicine to link certain complex phenotypes to specific region of the chromosome. It can help us to find the map of chromosome map on chromosome for a particular gene and its marker. The goal is the precise yeah, and it can be used to identify the action, interaction and number of location of the regions. The linkage analysis and association mapping are the two most commonly used methods for QTL mapping, and these exploits molecular markers in plant breeding, involves finding a subset of marker associated with one or more QTL that regulates the expression of complex traits. Currently, many QTL mapping are identified, and these QTLs are explained at a significant proportion of the phenotypic variance, and therefore give a rise to an optimistic assessment for the prospects of marker assisted selection. So this is very important for the marker assisted selection. We can easily identify the markers for a typical gene of interest, which is very much beneficial for a crop improvement. Students, I think this will be helpful for you, and please go through carefully this. If any doubt is there, please feel free to contact me. Thank you.